Hi, my name is Grant Kramer, and I am a professor emeritus at the University of Nevada, Reno. Welcome back to my series on plant physiology. Today we'll be talking about gibberellins. Plant Physiology, Lecture 15, the gibberellins, regulators of plant height. The gibberellin hormone was first recognized by Aichi Kurosawa, a Japanese scientist, in 1926 as a substance from the fungus Gibberella fujikiroi, which caused rice plants to elongate rapidly and fall over, which is called lodging, because the stems were too thin from rapid growth to support the rice panicles. Additional Japanese scientists successfully extracted gibberellin. It wasn't until after World War II that this discovery was made known to the rest of the world. It wasn't until 1958 that the first plant, gibberellin, GA1, was identified in runner bean seeds. It is now known that gibberellins consist of a family of over 130 diterpene compounds. The gibberellin hormones are numbered in order of their discovery. There are 4 known bioactive forms of gibberellin. They are GA1, GA3, GA4, and GA7. All other forms are either precursors or inactive forms. Gibberellins have a very significant physiological effect on plants in a multitude of ways. Gibberellins increase the height of plants by increasing the internode length between leaves. Gibberellins promote seed germination. They can induce flowering in some plants and affect fruit development. They can alter plant architecture in the shoots and roots of the plants and are now known to enhance nutrient uptake in roots under specific mineral nutrient conditions. Let's briefly examine these actions further. In this picture, you can see the effect of applying exogenous GA1 to dwarf and normal corn plants. GA1 causes dramatic increases in growth on the dwarf, but only minimal effects on the normal corn. Cabbage is a long day plant and doesn't flower until it is exposed to short days. Application of GA3 to cabbage under long days causes the plant to flower with large elongated flowering stalks. This elongation is commonly known as bolting. Applications of GA3 to ivy causes a reversion of the adult form of ivy to the juvenile phase, as seen here. Note the differences in leaf morphology here. In other plants, such as conifer trees, GA promotes the transition from juvenile to adult phases, so plant responses to GA is complicated. Gibberellin can influence sex determination, although its effects vary depending on the plant species, often promoting male flower development in some species like cucumber and female flower development in others like maize. In grapevines, GA inhibits flower development, which results in tendrils in place of flowers. In this figure, you can see in the lower panels normal tendril development in grapes. In the upper panels, the tendrils turn into flowers, which then form fruit in the GA-deficient grape mutant. Gibberellins promote seed germination. In this example, the radical emergence from the seed after imbibition in water of a gibberellin-deficient Arabidopsis mutant was observed initially at 18 hours after GA4 treatment, whereas the control seeds did not germinate. Wild-type seeds germinated after 24 hours. Gibberellins induce the growth of Thompson seedless grapes. The seeds are a source of gibberellin in grapes. The untreated seedless grapes on the left are naturally small. Spraying the grapes with gibberellin, the grapes seen here on the right, leads to the development of larger grapes and larger pedicels or grape stems. Applications of gibberellin stimulate the leaf sheath elongation of rice. So let's investigate further the physiological and molecular mechanisms for these actions. Let's start with the control of gibberellin concentrations. First, we'll discuss the biosynthesis and metabolism of gibberellins. The gibberellin biosynthesis pathway is a complex process taking place in 3 different cellular compartments: the plastid, the endoplasmic reticulum, and the cytoplasm. Gibberellins are composed of 5 carbon isoprene units, which are synthesized in the beginning of the pathway into geranyl diphosphate, shown here as GGPP. This pathway involves the terpene synthases, N-copolyl diphosphate synthase, or CPS, and N-choline synthase, or KS, which are involved in the synthesis of a key intermediate, N-choline. N-choline is oxidized by N-choline oxidase, or KO, and then by N-chorinoic acid oxidase, KAO, to produce Ga12 aldehyde, and eventually the aldehyde-containing intermediate, Ga12, in the endoplasmic reticulum. The bioactive GAs, Ga4 and Ga1, are formed in the cytoplasm in 2 different branching pathways, the non-13 hydroxylation pathway and the 13 hydroxylation pathway. From Ga12, the 2 parallel pathways form the final active GAs in the cytoplasm. A 13 hydroxylation step converts Ga12 to Ga53, initiating the production of the active GAs through additional oxidation steps by Ga20 oxidases and Ga3 oxidases. These steps ultimately lead to the formation of the bioactive GAs Ga4 and Ga1. Ga4 is 1,000 times more potent than Ga1. Ga2 oxidases further oxidize these bioactive forms into inactive forms, the Ga34 and Ga8. The GA biosynthesis pathway was largely elucidated by using mutants of Arabidopsis, P, and maize that were blocked in specific steps of the GA biosynthesis pathway. Many of these mutants were dwarf mutants that were easy to identify. In this figure, you can see each of the mutants associated with different steps in the pathway. Mutants are identified by lowercase names in italics. Green names are for Arabidopsis mutants. Blue names are for P, and orange names for maize mutants. Here you can see the impact of GA on plant size. In the left figure, you can see the wild-type plant versus each of the Arabidopsis mutants along the GA biosynthesis pathway. In the right figure, you can see the impact of increasing overexpression of the Ga2 oxidase in rice, which inactivates the bioactive GAs, resulting in smaller plants. In addition to Ga2 oxidation, gibberellins can be inactivated by glycosylation. The glycosylated form can be deglycosylated as well, making active GA. So this reaction is a reversible reaction. So there are many ways to control the concentration of bioactive GAs in the plant. Gibberellin concentration can also be controlled through GA transport processes. We know that gibberellins can be transported through the plant via the phloem. We have identified a number of transporter families that are capable of transporting gibberellins, as well as other molecules. They include some of the NPF family transporters, including MPF3 and the sweet family transporters, sweet 13 and sweet 14. Other sweet family transporters are known to be transporting sugars. In addition to measuring gibberellin concentrations by chemical means, such as LCMS, we can also visualize gibberellin concentrations in situ. 2 methods shown here are the use of a GA1 promoter Gus gene fusion in Arabidopsis that shows gibberellin concentrations in blue. On the right, fluorescently labeled GA was constructed to be able to better quantify GA directly in different tissues and cell types. You can read more about this in the paper by Shawnee et al. In the Proceedings of the National Academy of Sciences, listed here. So what are the physiological mechanisms involved in GA-induced growth? Here we can see the kinetics of GA-induced growth, which will give us some clues. If we excise leaf internodes to remove them from their source of GA, we can see the application of GA3 strongly increases the growth of the internode after about 4 hours. So this is a different longer growth response compared to the longer growth response of auxin-induced growth. Another way that growth is increased by gibberellins are that they stimulate cell division in the shoot meristem. Applied GA increases the number of mitotic divisions as shown here. Gibberellins increase the extensibility of the cell wall. They also lower the yield threshold of the cell wall, thus loosening the cell wall and accelerating turgor-driven growth. The cell wall space is not acidified as it is with auxin. However, there are increases in two cell wall loosening enzymes, expansins and xyloglucan transglucosylases. What is interesting is that the presence of auxin is necessary for GA-induced growth, and auxin stimulates GA biosynthesis. Again, note that GA effects on growth take much more time than auxin-induced growth. Here are a couple of figures showing the impact of auxin, or IAA, on gibberellin concentrations. On the left, we can see that applied IAA increases the concentration of GA1 in decapitated pea plants. On the right, we can see that IAA blocks the inactivation steps of GA1, thus stimulating GA1 concentrations and growth. Another way that plants control gibberellin biosynthesis is with the expression of the NOX gene. The NOX transcription factors directly suppress GA biosynthesis genes in the meristem, thus maintaining low GA concentrations in the meristem. This NOX-GA regulatory module is crucial for balancing cell division and differentiation in the meristem. It helps to maintain the meristem and not allow it to turn into an elongation zone. One of the other effects of gibberellins is on the promotion of seed germination. This process has been extensively studied in barley seed, largely due to the efforts of Russell Jones at the University of California, Berkeley. I had the pleasure of spending a 6-month sabbatical in his lab. His research lab has elucidated the process in quite a bit of detail. Barley seed germination starts when seeds are exposed to and imbibe water. During this process, gibberellins are synthesized in the embryo and released into the starchy endosperm via the scutellum. Once there, the gibberellins diffuse throughout the endosperm and into the alurone layer in the outer edges of the endosperm. Gibberellins induce the synthesis and secretion of alpha amylases and other hydrolases. Alpha amylase is an enzyme involved in the breakdown of starch into glucose. These sugars and other molecules are absorbed by the scutellum and transported to the growing embryo. Here we can see that GA stimulates the synthesis of alpha amylase by stimulating the synthesis of mRNA or messenger RNA of the alpha amylase. Gibberellin stimulates GA-MIB mRNA and subsequently alpha amylase mRNA by initiating a signaling cascade that ultimately leads to the transcriptional activation of the alpha amylase gene via the GA-MIB transcription factor. GA triggers the production of GA-MIB protein, which then binds to the promoter region of the alpha amylase gene, activating its transcription to produce alpha amylase mRNA. This is a critical pathway in plant seed germination where alpha amylase mobilizes stored food reserves. The entire process is summarized in this figure. Starting at 1 at the top of the page, GA1 from the embryo enters an alurone cell. Once inside the cell, GA1 may initiate a calcium calmodulin-dependent pathway necessary for alpha amylase secretion. 3. GA binds to GID1, which is a soluble GA receptor in the nucleus. 4. Upon binding GA1, the GID1 receptor undergoes an allosteric change that facilitates its binding to a DELA protein. 5. Once the DELA protein has bound the GA1-GID1 complex, an F-box protein, which is part of the SCF complex involved in protein degradation, is now able to polyubiquinate the GRAS domain of the DELA protein. 6. The polyubiquinated DELA protein is degraded by the 26S proteasome. 7. Once the DELA protein has been degraded, transcription of the GA-MIB gene is activated. The mRNA leaves the nucleus and is transcribed into a protein in the cytoplasm. 8. The newly synthesized GA-MIB transcription factor enters the nucleus and binds to the promoters of the alpha amylase and other genes encoding hydrolytic enzymes. 9. Transcription of these genes is now activated. 10. These mRNA leave the nucleus, are transcribed into enzymes on the rough endoplasmic reticulum, and packaged into secretion vesicles by the Golgi body. 11. These proteins are then secreted outside the cell by exocytosis to participate in starch degradation in the endosperm. The Green Revolution occurred in the mid-20th century and resulted from scientific advances in agriculture, including the development of high-yielding crop varieties, increased use of synthetic fertilizers, pesticides, and mechanized farming equipment. Spearheaded by scientists like Norman Borlaug, it aimed to increase food production to avert mass starvation in developing countries, significantly raising crop yields, especially for wheat and rice. Wheat and rice yields more than doubled during this period. A primary contributor to the Green Revolution was the development of new wheat and rice varieties with reduced stem height to prevent lodging or the falling over of the grain heads on the ground, or they would rot. The manipulation of GA concentrations through the breeding of GA genetic mutants was primarily responsible for the success of the Green Revolution. On the right side, there are images of how gibberellin research is being used to extend the Green Revolution through understanding and manipulating internode elongation, leaf growth and leaf angle, tassel and panicle branching, tiller development, and nutrient use efficiency. The effects of gibberellins on seed germination, plant growth, architecture, and nutrient absorption are profound and far-reaching. Wow, aren't the gibberellins a fascinating family of hormones? In this lecture, we talked about some of the important actions of gibberellins, particularly growth, elongation, and seed germination. I explained how gibberellin concentrations are regulated by biosynthesis, degradation, and inactivation. Gibberellin actions occur by altering gene expression in plants in substantial ways. Well, that's it. Stay tuned for future videos of plant physiology lectures, videos about grape growing, and winemaking by subscribing to my YouTube channel. And if you like this lecture, please like it on my YouTube channel so it will bring it to the attention of other people who might be interested in this subject. Have a great day.